Gram-positive endospore-forming bacteria. Gram-positive spore-forming bacteria. That is what we are addressing today. We are looking at the gram-positive endospore-forming bacteria, and because spore is the same thing as endospore, we can also call them gram-positive spore-forming bacteria. What are the importance of bacterial endospores? That is what we want to look at first before we go into the gram-positive spore-forming bacteria fully. We have to understand what the importance of bacterial endospores are. Bacterial endospores play an important role in survival and dissemination of bacteria. Endospores are highly resistant structures that form within the cells of certain bacteria, providing a means for survival under adverse conditions such as extreme temperatures, desiccation, and high levels of environmental stress. The robust nature of endospores enables them to persist in the environment for extended periods of time, providing a source of bacterial inoculum for new growth and colonization. In addition, endospores can be found in the soil. It can also be found in water and other environments, and they have the ability to infect plants, animals, and humans, making them important in the spread of infectious diseases. In biotechnology, endospores are useful as model systems for studying cellular physiology, genetics, and biochemistry. Endospores can be easily preserved, stored and transported, making them valuable resources for research in various fields, including biochemistry, microbiology, and biotechnology. They are also utilized in industrial applications, such as food preservation and sterilization processes. Bacterial endospores play an important role in the survival, dissemination, and spread of bacteria, and they are valuable resources in various fields of study and industrial applications. Now, let's move on to the process of spore formation. Spore formation is a survival strategy used by certain bacteria to protect themselves from adverse environmental conditions such as extreme temperatures, desiccation, ultraviolet radiation, high salt concentrations. Spores are highly resistant to environmental stress, and they can remain viable for extended periods of time. When conditions improve, spores will germinate into active cells and they will resume growth. Bacterial spores can play an important role in the survival and dispersal of bacterial populations, allowing the bacteria to colonize new environments. They are also a common cause of food spoilage, and many pathogenic bacteria are capable of producing spores, making them a significant threat to public health. Spore formation is a complex process that involves the accumulation of protective layers around the cell and the cessation of cellular metabolism. The mechanisms of spore formation vary among different bacterial species, but they all serve the same basic purpose of providing protection to the cell during adverse conditions. Bacterial spores play a significant role in the survival and dispersal of bacteria, both in the environment and within hosts. Understanding the mechanisms of spore formation and the conditions that trigger it is important for developing strategies to prevent the spread of pathogenic bacteria and for controlling spoilage food spoilage. Bacteria that form endospores and are gram-positive. Representative genera of gram-positive spore-forming bacteria include Bacillus, Clostridium, Micromonospora, Saccharopolyspora, and Streptomyces. The gram-positive spore-forming bacilli are the Bacillus and Clostridium species. These bacilli are ubiquitous, and because they form spores, they can survive in the environment for many years. Bacillus species are aerobes, and the Clostridium species are anaerobic. Of the many species of Bacillus and related genera, most do not cause disease and are not well characterized in medical microbiology. There are a few species, however, that cause important diseases in humans. Anthrax, a classical disease in the history of microbiology, is caused by Bacillus anthracis. Anthrax remains an important disease of animals and occasionally of humans because of its potent toxins. Bacillus anthracis is a major potential agent of bioterrorism and biological warfare. Bacillus cereus and Bacillus thuringiensis cause food poisoning and occasionally eye or other localized infections. The genus Clostridium is extremely heterogeneous, and more than 200 species have been described. The list of pathogenic organisms as well as novel species isolated from human feces, whose pathogenic potential remains undetermined, continues to grow. Clostridia cause several important toxin-mediated diseases, including tetanus, that is caused by Clostridium tetani, botulism, that is caused by Clostridium botulinum, gas gangrene, that is caused by Clostridium perfringens, and antibiotic-associated diarrhea and pseudomembranous colitis, that is caused by Clostridium difficile. Other Clostridia are also found in mixed anaerobic infections in humans. The genus Bacillus includes large aerobic gram-positive rods that occur in chains. The members of this genus are closely related, but they differ both phenotypically and in terms of pathogenesis. Pathogenic species possess virulence plasmids. Most members of this genus are saprophytic organisms, prevalent in soil, water, and air, and they are also found on vegetation, such as Bacillus subtilis. Some species of Bacillus are insect pathogens, such as Bacillus thuringiensis. This organism is also capable of causing disease in humans. Bacillus cereus can grow in foods and has been implicated in food poisoning. They can grow in foods and cause food poisoning by producing either an enterotoxin, in the case of diarrhea, or an emetic toxin, as in the case of vomiting. Both Bacillus cereus and thuringiensis may occasionally produce disease in immunocompromised humans. Now, I must say here that apart from the causing of disease or being pathogenic, these organisms, especially Bacillus, is well known as a biodegrader in the field of in the area of biodegradation. It's a well-known degrader. Most Bacillus subtilis and some other species of Bacillus which are not pathogenic have been known to break down hydrocarbon, move it from complex molecule to its simpler molecules. So we are talking about the ability of Bacillus cereus and Bacillus thuringiensis to generally produce disease in immunocompromised humans. This is talking about normally where they will not cause disease, but because the human is immunocompromised, the immunity of the human has dropped. So these organisms are able to cause disease in such a case. Example is meningitis, endocarditis, endophthalmitis, and conjunctivitis or acute gastroenteritis. Bacillus anthracis, which cause anthrax, anthrax is the principal pathogen of the genus. So anthrax is a deadly disease, a fatal disease. It's a disease that leads to death in most cases, and therefore Bacillus anthracis is the principal pathogen of the genus. Now let's look at the morphology and identification of this organism Bacillus. Typical organism. The typical cells measuring 1 × 3 to 4 micrometer have square ends and are arranged in long chains. Spores are located in the center of the bacilli. I've shown you the different type of spores already in this course, where I talk about the different locations of the spores in the cell. Some can be at the center, some at the end, and we have looked at that already. Culture media. Colonies of Bacillus anthracis are found around and have a cut glass appearance in transmitted light. Hemolysis is uncommon with Bacillus anthracis, but it is common with Bacillus cereus and the saprophytic bacilli. Gelatin is liquefied, and growth in gelatin stabs resembles an inverted fig tree. Growth characteristics of the organism. The saprophytic bacilli use simple sources of nitrogen and carbon for energy and growth. The spores are resistant to environmental changes, and they withstand dry heat and certain chemical disinfectants for moderate periods. They persist for years in dry earth. Animal products contaminated with anthrax spores, e.g. hides, blisters, hair, wool, bone, can be sterilized by autoclaving. The Clostridia are large anaerobic gram-positive motile rods. Now we have moved on to Clostridium. Many Clostridium decompose proteins or form toxins, and some do both. Their natural habitat is the soil, marine sediments, sewage, or the intestinal tract of animals and humans, where they live as saprophytes. The Clostridia continue to increase in number as new species are discovered and several species have been sequenced. There are 19 clusters based on 16S ribosomal RNA gene sequence analysis. Most clinically related species are in RNA cluster. 1. Among the pathogens in this cluster are the organisms causing botulism, tetanus, gas gangrene, and pseudomembranous colitis. Now we'll move now to the morphology and identification of Clostridium. Typical organisms. Spores of Clostridia are usually wider than the diameter of the rods in which they are formed. In the various species, the spore is placed centrally, subterminally, or terminally. Most species of Clostridia are motile and possess peritrichous flagella. A gram stain of the Clostridium species with terminal spores. Clostridium gram stain. Individual gram-positive bacilli are present. Many are in chains. Some of the bacilli have spores, which are the unstained or clear ovoid shaped arrows. So this is showing the gram stain reaction of Clostridium as seen under the microscope. Culture. Clostridia are anaerobes. They are anaerobic and they grow under anaerobic conditions. A few species are aerotolerant. That means they can grow in the presence of oxygen, but they just tolerate oxygen, not that they grow strictly or perform well in the presence of oxygen, and also grow in ambient air. In general, the Clostridia grow well on the blood-enriched media or other media used to grow anaerobes. Colony forms. Some Clostridia produce large raised colonies, e.g. is Clostridium perfringens. It produces large raised colonies. You remember I told you the other time that under the plate, petri dish plate, you can characterize bacteria to certain extent. So you can see here now it's talking about the colony, whether it is raised or otherwise. So in the case of Clostridia, some Clostridia produce large raised colonies, e.g. Clostridium perfringens. Others produce smaller colonies, e.g. Clostridium tetani. Some Clostridia form colonies that spread or swarm on the agar surface. Clostridium septicum. Many Clostridia produce a zone of beta hemolysis on blood agar. Clostridium perfringens characteristically produces a double zone of beta hemolysis around colonies. Growth characteristics. Clostridia can ferment a variety of sugars, saccharolytic, and many can digest proteins, proteolytic. Some species do both. These metabolic characteristics are used to divide the Clostridia into groups. Milk is turned acid by some and digested by others, and undergoes stormy fermentation, that is, clot turned by gas, with a third group, e.g. Clostridium perfringens. Various enzymes are produced by different species. Clostridium species produce Clostridium species produce more toxin than any other group of bacteria. So they produce more toxins than any other group of bacteria. I would like you to read more on Clostridium toxin because they produce more toxin than other group of bacteria. Read more about the toxin produced by Clostridium. We are going to continue this class after this break.